Hi, my name is Dear Tiagi, and in this video, I'll be going over AP Bio Unit 3, Cellular Energetics. So to start, metabolism is a key concept in this unit, and the idea is that metabolism is the sum of all reactions within a cell or organism. So if we consider a metabolic pathway, right, we're going to have a starting product, which is then going to an intermediate via reaction, and multiple intermediates, right, so we have multiple reactions. And then eventually the sum of all these reactions, the sum of this metabolic pathway leads to a final desirable end product. And so there are two types of metabolic pathways that we categorize. We categorize them as catabolic. Catabolic pathways release energy by breaking molecules, and anabolic absorb energy by building molecules. Okay? And so within these reactions, an important concept is that we use enzymes, which are biological proteins that help to speed up the reactions. So they're called catalytic proteins because the idea is they catalyze reactions. So a reaction, if we consider a energy versus reaction rate graph, a reaction like so, right? Here we have the energy of the reactants and products. Reactions require a certain level of activation energy to start, right? In order for this reaction to occur, to get to the products, we need to get enough energy to reach here in order for them to happen. So what enzymes do is, while they don't change the amount of energy change, which is denoted delta G, or free energy, they do change the activation energy required. They reduce this activation energy. Okay, and by reducing it, it takes less energy for it to become, for the reaction to occur, and therefore it speeds up the reaction. And so we have two types of reactions in chemistry. We have endergonic reactions, which are similar to anabolic pathways because they absorb energy to occur. And we have exergonic, which you can see from the word X, releases energy, which are similar to catabolic pathways. So an enzyme, if we look at it specifically, has a certain shape, and then we have these reactants, right? The reactants are called substrates, and these substrates, in order for the enzyme to work, bind to the enzyme in the place called the active site. And so in order for the enzyme to actually bind with the substrate, the enzyme has this thing called induced fit, where it has a certain shape and charge in order to induce a fit with the substrate. So it's kind of like a key. So it has a certain shape, so it can induce a fit with the substrate. So what affects the efficiency or the rate of reaction? So enzymes, what affects enzymes, which then affect the rate of reaction, we have temperature, pH, and for temperature, pH, there's an optimal range for enzymes to occur, for the enzymes to work, right? And outside of this optimal range, if we deviate from it, right, if we have a too hot of a temperature or too cold for that specific enzyme, what happens is we get denaturation. So you should know that proteins, if they're outside of a range, start to denaturize or break down into forms that don't work. And so this denaturization prevents enzymes from working if they're outside their optimal temperature or pH. Additionally, the concentration of enzymes / substrates all serve to affect the reaction rate of a reaction. And enzymes, what they do also, they have this thing called negative feedback. So the idea is when negative feedback, enzymes, when they finish their product, they get the end product, their end product prevents the starting product from happening, right? It prevents an enzyme here from occurring, which then prevents this whole pathway from occurring. And so why would a molecule or a cell want to do negative feedback? Well, the idea is we don't always want this end product to be made. And so the end product decides, okay, let's make this molecule that prevents the reaction from occurring. And so therefore, we're not wasting energy or resources into making a molecule we don't need anymore. And so with negative feedback, how it works is we use two types of negative feedback for enzymes. We use competitive inhibition to stop enzymes, and we use non-competitive or allosteric inhibition. So competitive inhibition, if we imagine an enzyme, right? So if we imagine an enzyme, let me just erase all this quickly. Competitive inhibition involves the thing that's stopping the enzyme binding to the active site. So competitive, it binds directly to the active site, and this prevents the substrate from binding, right? Because the enzyme is already being binded by the competitive inhibitor, and therefore it can be binded. Non-competitive, on the other hand, binds to another section of the enzyme, but then it changes the shape, right? So it changes it to some kind of weird shape. And again, like we said before, it needs a specific shape for the substrate to bind. So the substrate cannot bind, even though it's not being competitively inhibited, because it's a weird shape and it can't bind to that. Next, let's quickly go over the laws of thermodynamics. So the first law of thermodynamics states that energy of the universe is constant. So you can create it or destroy it, which means that we can make it or create it in any of our reactions, biological reactions. And the second law states that entropy is increasing in the universe. So entropy is a measure of disorder, and the way this relates to biology is that whenever we have a reaction, we always lose energy in some sort of way. We can lose it in terms of heat or in terms of sound, but we always lose energy whenever we have a reaction. Okay? And so what cells do, right? So cells, again, the endergonic and exergonic, cells use this process called energy coupling to mitigate the fact that energy is constant. So energy coupling involves having a exergonic process, right, that releases energy, and pairing it with an endergonic process. So we use the energy we start from the exergonic process to then start the endergonic process. And the idea is this energy is usually in the form of ATP, and we break this ATP into ADP plus P, right? And this then makes energy, which we use for the endergonic reaction. And this is called energy coupling, which is a process that cells do to kind of mitigate the fact that energy is constant and cannot be created or destroyed. Next, let's go into photosynthesis. So photosynthesis is the process by which autotrophs, which make their own food / energy, make energy. So photosynthesis, right, occurs in the leaves, and the reason why the leaves are green in a plant is because of chlorophyll. So chlorophyll is green because it reflects green light. So photosynthesis works best under blue and red light because that's what most chlorophyll in plants like to absorb. Right, so chlorophyll consists of pigments that absorb different wavelengths of light, and again, the wavelengths it likes to absorb are red and blue. And photosynthesis is a anabolic pathway. And the formula for photosynthesis, which you should probably know, is 6CO2 plus 6H2O plus obviously light yields glucose, C6H12O6 plus 6 oxygen. So this is a redox reaction, which we can see, right? Because what's happening is that the carbon dioxide is being reduced, so it's gaining electrons, right? Reduction gains electrons, oxidation loses electrons, while the H2O is being oxidized, right? It's losing the H, so it's losing its electrons to become oxygen. So there's a redox reaction there. And so the CO2 and oxygen, they're both gases. They enter and exit via the stroma. So photosynthesis, important thing to know, is that it occurs in the chloroplast. So photosynthesis occurs in the chloroplast, and inside the chloroplast, we have these kind of sacs called thylakoids. And the thylakoids can be arranged in these things called grana, right, stacks, and that's where photosynthesis occurs, and then you have the stroma as well. So photosynthesis occurs primarily or fully inside these chloroplasts, so it's self-contained. Okay, and looking at the reaction specifically, right, so we imagine the chloroplast, it all happens in the chloroplast. We have our H2O as an input, and its input's first used in the light reactions. So light reactions take in energy from the sun, and the output of this light reaction is oxygen through a process called photolysis, where the hydrogen or the water is split into oxygen. This light reaction then creates ATP, and it creates this molecule, it oxidizes the molecule into NADPH, and this oxidized molecule then goes into the Calvin cycle, which is a self-regenerating cycle that affixes CO2. So we have another CO2 input, and it asphyxiates it into glucose, which is our output, right? And then this uses energy. Energy is in the form of ATP, and it breaks that energy into ADP, which then returns to light reactions. So we have ADP plus P, and it also uses the electron that we carried in NADPH, and we have NADP plus returning. So NADPH is called a electron carrier, and the idea is it serves to carry electrons, right? When it's in this form, NADP plus, it's empty, you can think of, and then when we had light reactions, we affix an electron, we oxidize it, we reduce it into NADPH, and we give it an electron, which is then used in the Calvin cycle. So the main idea you need to know for these is just the location of where each cycle occurs, and mostly in the inputs and outputs of each stage. So the first stage is light reaction, right, which occurs in the thylakoid and the thylakoid membrane. So this is the thylakoid. And so what we have is we have two photosystems here. And a photosystem is just a section in the membrane with these pigments, chlorophyll, right? And what happens is we first have water that goes inside this thylakoid, and this water is split through photolysis into oxygen and to electrons, right? And these electrons go inside the photosystem. And then what happens is we have this energy through light into the sun that hits the photosystem. And what this does is it excites the pigments and the electron, which is then carried into the electron transport chain. So the electron transport chain starts with a lot of energy, and then it goes from the electron transport chain to photosystem 2. And while it's going, it loses energy through oxidation. And this loss of energy then allows for these hydrogen protons, or protons in general, to enter the thylakoid through active transport, right? Because these things, or these protons, are charged, so they need to go through active transport. And so the energy we get from this electron going through the electron transport chain, right, through the thylakoid membrane, allows for these protons to be pumped inside. Because again, this is also inside, we have a high concentration of protons, low concentration of protons outside. Not the best place to put it. So we need to use active transport to pump in the protons, which we do with the energy from the electron transport chain. Photosystem 2 then absorbs more energy, which then powers the electron transport chain again. And at the end of the electron transport chain here, we have a enzyme, right? So an enzyme that serves to fix NADP plus and a proton into NADPH. So we have an electron carrier now. And one of the things is by pumping in these protons, right, we have a high concentration of hydrogen or protons inside. We have this enzyme called ATP synthase, which is kind of like a little motor. And what the particles do is this high concentration, serves to then exit through this ATP synthase. So it exits through here, and while it exits, what happens is we have this ADP and phosphate outside, which we then use the energy from it exiting through ATP synthase to create ATP. this ATP and this NADPH then both go to the Calvin cycle. And then in the Calvin cycle, you really don't need to focus a lot on specifics, but the idea is with the Calvin cycle, we start with a 5-carbon, we should erase a little bit, but we start with a 5-carbon molecule called RUBP. We then get our CO2, which makes it a 6-carbon molecule. We split it into two 3-carbon molecules. We then use our ATP and our NADPH, which then of course become ADP and NADP plus to create G3P. G3P is then used to make glucose, right, which is one of our final products. And we also then have 5 G3Ps left. These 5 G3Ps are then made back into RUBP. And this occurs, the Calvin cycle, in the stroma. And there's 3 stages. We have carbon fixation, where we fix the carbon together. Then we have reduction, where we reduce it into glucose. And finally, we then have the regeneration stage of RUBP. But again, the main idea is just to know the location is the stroma and the inputs and outputs. Carbon's the input, output is glucose. The intermediaries aren't really important for the Calvin cycle in this course. And finally, we'll go over cellular respiration, which is a catabolic pathway. And the idea is with cellular respiration, we take a organic compound and we convert it into ATP. So the cellular respiration is basically the opposite of photosynthesis in terms of the reaction and the steps really. We have 6CO2 plus C6H12O6. Oh, sorry, 6O2, not CO2. That's for plants. This yields 6H2O, so water, plus 6CO2 plus energy in the form of ATP. Okay, and again, this is a redox reaction as well. It's catabolic as opposed to anabolic from before. And so here we have the oxygen, which is now being reduced, right? So it's gaining electrons. And then we have the glucose, which is being oxidized because it's losing its hydrogen, so it's losing its electrons. And just the main steps of cellular respiration in general, we have glycolysis, which occurs in all cells, right, which suggests a common ancestor. And glycolysis, what it does is it occurs outside in the cytoplasm, right? So we have the mitochondria here. It occurs outside in the cytoplasm, and what it does is it takes glucose and converts it into pyruvate. Okay, and that's what it does. It also creates some ATP, but not too much ATP, just a little bit. And then we enter into the mitochondria. And the first step we have is pyruvate oxidation, which then takes this pyruvate and turns it into acetyl-CoA. Then after pyruvate oxidation, we have the Krebs cycle, which is pretty similar in terms of to the Calvin cycle. Again, you don't really need to know the intermediaries for it. You just need to know that the idea is what it does is it takes the acetyl-CoA and it creates these carriers. And then we go into the final step, which is called oxidation phosphorylation. And we also create ATP in this Krebs cycle step as well. And so the ATP we're creating in these 2 steps is through substrate phosphorylation, right? But in the final step, what we create is we create ATP via oxidative phosphorylation. So phosphorylation. So the idea is with this phosphorylation, we take a ADP plus a P, we add energy, and we get ATP, which is our energy molecule. With substrate phosphorylation, the phosphate is just freely floating in the stroma, right? Or it's not the stroma, it's freely floating just around. Or so no, it's the phosphate, sorry, I mixed it up. The phosphate is from the substrate, right? But then oxidative, the phosphate is from the just freely floating around, okay? And so through oxidative phosphorylation, we use our carriers and we use the energy we've created a little bit to then create a lot of ATP. And the majority of our ATP is created through this final step. And both pyruvate oxidation and Krebs cycle occur in the mitochondria matrix, so inside of like the cytoplasm of the mitochondria, versus oxidative phosphorylation, which involves the electron transfer chain, occurs inside the membrane, inner membrane of the mitochondria. So it's kind of the opposite steps as the photosynthesis, right? Because we end with the electron transfer chain. So it's kind of the opposite. Okay. So again, the important idea is that we have 4 steps, right? I got glycolysis, which is 2 sugars in, and we again have these carrier molecules that are made throughout. So we have NAD plus here, and we have FAD plus. And these carrier molecules, again, these are empty, but then throughout the process, we make them into NADH and FADH2. These carried electrons, which are then used for oxidative phosphorylation. And just to go over that process quickly, the idea is with oxidative phosphorylation, we have, again, an electron transfer chain. We pump in the protons, right, into the space, which then power ATP synthase. And then these carriers are what provide our electrons, which we use for the electron transfer chain, which then pumps in, again, the protons, which helps power ATP synthase. And finally, we also have, for organisms that don't use oxygen, we have anaerobic respiration, which is respiration without oxygen, right? So if for regular cellular respiration, at the end of the electron transfer chain, the electron is fixed to oxygen and becomes H2O, right? But without this oxygen, if we don't have it, we can't do this process. So we can't do oxidative phosphorylation, and instead we have to do fermentation, and which means we can't use oxidative phosphorylation, and instead we have to use substrate phosphorylation, which creates much less ATP, but it works without oxygen, which is why we do it. And there's 2 types. There's alcoholic fermentation, right? And the idea is we take the pyruvate, right? And we need to convert it into something and it becomes alcohol. Or in humans, we have lactic acid fermentation. So we take the pyruvate from glycolysis and we change it into lactase, which builds up in our muscles and is eventually then, just waste product. And yeah, that's all you need to know for AP Biology Unit 3. Thank you all for watching, and I'll see you guys all tomorrow. Bye.